It's my pleasure to introduce Alexis Jackson. She's an intern in the plant ecology program at Archbold, supported by the Vaughn Jordan Foundation. Alexis is originally from Georgia and got her BS in biological sciences from Virginia Polytechnic Institute, VPI, where she did an independent research project measuring carbon dioxide and methane emissions, along with changes in inundation and dissolved organic carbon in riparian wetlands. She also is a mentor in their life sciences mentoring program at VPI. She's been an intern here for several months, but is leaving a little bit earlier than usual because she has a great opportunity to begin attending the University of Florida in the environmental education and engineering program under David Kaplan's Watershed Ecology Lab. AJ has been a great intern for us and has tremendous enthusiasm and has taken advantage of natural gradients in the landscape at Archbold to look at patterns of biomass in a number of species along that gradient. So it's my pleasure to introduce Alexis.
Hello, everyone. Thank you, Eric, for that introduction. Hi, everyone. My name is Alexis Jackson. I am the Vaughn Jordan intern here in the plant ecology program at Archbold Biological Station. And I'll be presenting today my research that explored the above ground biomass variation along elevation gradients in the Florida scrub. Okay, so what made me interested in this topic? I have a background in wetland ecology. My undergraduate work was looking in wetland biogeochemistry. I looked at carbon dioxide and methane emissions coming from ephemeral wetlands. Therefore, I did have knowledge on wetlands and how they function, but didn't take into account how they may affect plants. When being in the field at Archbold, I always noticed that there was thicker vegetation, mostly palmettos, nearest to the wetlands. And when I was in the middle of a rosemary scrub, there was nothing but rosemaries. So I wondered how plant biomass changed between these two completely different habitats, even though they're within mere meters of each other. I was also interested in seeing how elevation affects plant dynamics in the scrub, since this is such a unique area. So you can see pictures of me from undergrad. That's me with a flux chamber, taking the carbon dioxide and methane emissions. And then at the bottom, that is me when I was doing actually tadpole work with my old lab. So what is above ground biomass? Above ground biomass is the weight of the living plant material contained above ground surface. So this includes the stems, the leaves, the seeds, and the branches. And why am I observing it? Why is it important? Plant biomass helps us understand ecosystem structure, and especially post major disturbances, and can give us insight into dominance among species. As you can see in the figure on the right, it was part of a paper called the Association of Ecotones with Relative Elevation in Fire in Upland Florida Landscape. Excuse me. And this supported that across an elevation gradient, as you can see above, we have different habitats or plant assemblages, which are just the uniform groups of plants in a similar habitat. So we had median relative elevation along the x-axis, and 0 is the lowest elevation, and then 250 is the highest elevation. So at the lower elevations, you have flatwoods. In the middle elevations, you have scrubby flatwoods. And then at the higher elevations, we see the rosemary scrubs. So I wanted to measure above ground biomass in a non-destructive and time efficient way. So there have been studies that use regression equations to predict above ground biomass. And I used the Mingus and Smith paper from 2019, and where they estimated above ground biomass across like 14 dominant scrub species. Some of them are the same ones I'm using in my research today, across the same ecosystems that I surveyed, and in 5 different time since fire classes. So they got a good range of time since fire fields. And I used a regression equation and calculated coefficients to look at biomass across an elevation gradient. These were helpful because, again, they don't require me to uproot this plant in any way or cut down anything. It was non-destructive. Okay, so my main question was, how does plant biomass change in proximity to the water table? So I hypothesized that plant biomass would decrease as relative elevation increase because of distance from the water table and lower levels of nutrients available. So just to keep in mind that relative elevation along the x-axis is increasing from left to right. Therefore, in the flatwoods, I expected that there would be more biomass at lower elevations, and then scrubby flatwoods would be in the middle. And then the rosemary scrubs, which are at the highest elevations, would then have the lowest biomass. So now I'm going to talk about the 9 species of plants that I looked at. I looked at these 9 species of plants because they're the most dominant scrub plants along all of my transects and in my habitats. So first I looked at 3 different oaks: Quercus chapmanii, also known as Chapman's oak; Quercus inalpina, also known as scrub oak; Quercus geminata, also known as sand live oak. I looked at 2 vacciniums: Vaccinium mercenites, which is also known as shiny blueberry; Vaccinium darii, also known as Darrow's blueberry; 2 palmettos: Sable Antonia, a scrub palmetto; Serenora repens, which is also known as saw palmetto; and then 2 Lyonias: Lyonia lucida, which is also known as shining featherbush, and Lyonia fruticosa, which is known as coastal plain staggerbush. So to give some background about where we're located in the Lake Rose Ridge. That square at the southernmost tip of the Lake Rose Ridge is where Archbold Biological Station is. At the bottom is a map showing all 11 rosemary scrubs I surveyed and their corresponding wetlands I sampled on both of the south and north portion of the station. The rosemary scrubs criteria were selected based on time since fire. So I kept it consistent and stayed between 3 to 6 years. So they would burn last between the years of 2015 and 2018. My criteria for the wetlands were that they had to be interior, so not near the road, and of course had to be within 200 meters of a rosemary scrub. That was just so I wasn't running more than 200-meter transects. So just to give more background about what the habitats that I surveyed in. I surveyed in a rosemary scrub, which is an open area where mostly Florida rosemary is present. As you can see with Haley standing in the background, rosemary gets really big and there's not much else in there. From the bottom picture, you can see maybe 1 palmetto, maybe 2 or 3, but they're mostly dominated by Florida rosemary. Flatwoods, which are open stands of grasses / pine and palmettos. In the picture, you can see the pines, you can see the dense palmettos, and you can see plenty of grass. And then scrubby flatwoods, which is an open canopy of pine trees, shrubby oaks, and palmettos. So you can see this is way more dense than the last 2 habitats. All right, so here you can see a map of what a transect would look like. Pictured is bald 29 east and west, but we're just going to focus on west for this example. So shown in this is the dominant vegetation, which included rosemary being, of course, the most dominant in the rosemary scrub, and then wiregrass being most abundant in pretty much every other area. A transect was created on ArcGIS from the highest point in the rosemary scrub to the wetland edge. Then 10 points were randomly generated within this transect. I randomly selected 1 representative of my non-plant species at each point. So this totaled in like 110 sampling points across 11 bulbs. So to the right, you can see a picture of the transect that I actually set up. This is just half. Some of them are pretty long, so you couldn't see the whole extent from one picture, but this is just an example. And then to the left, you can see a picture of me sampling an oak or about to sample an oak. And this is just how I would take the length, width, and height of each of the plants. So this is just another oak that I was measuring the length of, the height of. And to keep it consistent, individual plants were defined as any stems within 15 centimeters of the initial stem observed. So now I'm going to be talking about my results. So first things first, I found the relative elevation, which is just the elevation difference between the wetland and bald or wetland at each point. So everything is relative to the wetland, to keep that in mind for the coming ups. Then I calculated the biomass using length, width, and height and inserting that into the biomass regression equation that I previously talked about from the Mingus and Smith paper. Then I ran a general linear model to see if there was a significant relationship between overall biomass and relative elevation. And as you guys can see from the first row, there was no significance. However, each individual species biomass and relative elevation had their own relationship, which was significant or not. As you can see, Quercus geminata was the only one that wasn't significant. So relationship between biomass and relative elevation was dependent on species. So to start off, I have Quercus chapmanii. We are viewing Quercus chapmanii's biomass plotted along relative elevation. In the bottom left, there's a picture of flatwoods in the wetlands. That's to help represent the lower elevations. And then in the top right, you guys can see a picture of a rosemary bald. And that is to signify the higher elevations. So Quercus chapmanii and geminata both followed a positive trend. So as their biomass increased, relative elevation increased. However, Quercus and opina did not. So here I have Serenor repens plotted along Serenor repens' biomass plotted along relative elevation. Again, I have a wetland picture in the bottom left, so you guys can get an idea of that is a lower elevation. And then in the top right, I have rosemary scrub to signify the higher elevations. So both of the palmettos had a negative trend. As relative elevation increased, they decreased, which means that they had a higher biomass in the lower elevations, the flatwoods, which was also observed. So here now I have Vaccinium mercenites. So the biomass again is plotted along the relative elevation. In the top, you can still see the picture of the rosemary scrub, and in the bottom, we see the flatwoods still. So for this one, Vaccinium mercenites did show a positive trend. However, there was not many samples or surveyed plants in this, and neither was it from Darwii. Therefore, these are loose trends. The R-squared values also weren't strong as well. So I think with more samples, this could have been a stronger trend, but mercenites did show a positive trend, and Darwii did show a negative trend. Okay, and then we have Lionia fruticosa. So you're still viewing the biomass plotted along the relative elevation. It had an overall negative trend, and Lionia fruticosa and Lionia lucida both had a negative trend. So as relative elevation increased, they decreased in size. Therefore, they were bigger towards the scrubby flatwoods and flatwoods habitat. So in conclusion, I did not find an overall relationship between biomass and relative elevation. However, I did realize that, again, this was mostly species specific and it was species habitat specific. So for the oaks, I realized that they had better optimum growth in the scrubby flatwoods, and that heavily is characterized by scrubby flatwoods. Oaks are big characteristics of scrubby flatwoods. Therefore, this should have been expected. The palmettos, again, are a big characteristic of flatwoods. So they had optimum growth in the flatwoods, which is why they increased. As relative elevation increased, they decreased in size. because their optimum growth isn't in rosemary scrubs. Same with the Lyonias. As relative elevation increase, they decrease in size. So they're also a characteristic of the flatwoods environment. So, and for vaccines, as I said before, with more samples, I feel like I would have had a stronger relationship and then could quantify that a little bit more. So for future direction, if I had more time to do this project, I would definitely look at a variety of times since fire. Some that maybe had burned closer to fire, so 1 to 2 years post-fire, and then some a little bit longer than 6 years, so 7 to 8 to 9. A lot of this ecosystem often has higher above ground biomass accumulation right after fire, so that would have been interesting to see. I would take observations of each habitat when I'm at the plant species, so I was something I could use as a factor in my actual results instead of just like observing afterwards. So that was a thought. I would explore other factors like soil nutrients and soil microbes to see if those are factors in plant biomass. And I would also survey more target plants. So instead of just doing like 10 transects or just picking 11 transects and just picking one species to survey at each point, I would pick maybe 2 palmettos to survey at each point, or 3, just so I was getting a good variety. Oh. And that is the end. I want to say thank you to everyone. I really appreciate you guys tuning in. I want to really say thank you to the Plant Ecology Program. Thank you to Eric, Stephanie, Lexi, Scott, Aaron, and Haley. Thank you to the GIS Lab for getting me that relative elevation data, that LiDAR data. Vivian and Ryan, I really appreciated that. And thank you for the Von Jordan Foundation for funding my internship. And also thank you to all the amazing people I've met here at Archbold. Do you guys have any questions?
Thank you, AJ. You did great. And we have, please submit some questions. We have a few questions. I'm going to start out with one of my own. In the paper you mentioned in the beginning by Betsy Boughton et al., they found kind of gradual changes in species distributions along that elevational gradient. And I'm wondering what your impression is of the changes in biomass and whether you feel like these flatwoods and scrubby flatwoods and rosemary scrub have very distinct boundaries in terms of biomass, or whether it's a more gradual change.
I would say it's more of a gradual change than more of like a distinct, like you can tell when. However, in that paper, they actually categorized the ecotones, which is like the space between the plant assemblages, which is like the habitat. They characterized that. I didn't do that in my paper, so, but I think that, yeah, it was more of a gradual change in biomass, especially from the flatwoods to the rosemary scrub, because those are completely different habitats, and you can definitely see that along any transect.
Okay, there's a question from Grace, and she asked, how did you acquire elevational data in your project? How did you get the relative elevation from the wetland?
Oh, so the relative elevation is just the difference between, like I said, the point and the wetland. So, you know, I got the data from like the LiDAR data, but how I calculate the relative elevation is just the elevation at each point minus the elevation of the wetland. So that was my relative elevation.
Okay, can you explain what the LiDAR data, how they collect that data?
Oh, I cannot explain that. I actually don't know how they collect that data.
Okay.
I know that they use, yeah, no, I actually do not know how they collect that LiDAR data. I know that at each point, I actually took GPS points of like what the elevation would be at each point, but I actually ended up using the GIS data just because it was more accurate. But no, I'm not sure how they accumulated that.
I can fill that in for, since I posed the question, is the LiDAR data is, there are pulses that from above that go down and bounce off either the ground or the vegetation. And the ones that bounce off the ground give you very precise elevational data. So it's actually taken from either satellites or aircraft across large areas, which is a really cool technology. Margaret Davenport has a question. She asks, how would you incorporate measuring soil nutrients into this research?
Oh yeah. So if I could do soil nutrients, I think I would, instead of how I did it on, well, I did not do soil nutrients for this, but for this project, I did like 10 sampling points across my transect length, which could be anything from like 30 meters to 150 meters. I probably would break it up by habitat and then do soil nutrients that way. I think it would be a little much of overkill to do a soil sample or soil nutrients at each sampling point. That would just be a lot of soil samples and it wouldn't say much, but I think I would see more of a change in each habitat, because obviously that's what we've seen in this study, that we see changes between habitat more than anything. So that's what I would do.
Okay. There's two related questions about vaccinium species. And David Paddock asked whether you had hypotheses about why the two vaccinium species show different trends. And Aaron Hotchkiss wanted to know whether you predicted these different trends of the two vaccinium species, and if you had an explanation, maybe in terms of plant traits or something else that might explain why they had different trends.
Yeah, so I think, well, for Vaccinium dorii, I think the problem with that one and why I saw that trend was just simply because I didn't have enough sampling, of that plant. So I didn't really have a hypothesis for that one, but for Mercenites, I definitely seen it going in that same trend. I can actually go backwards because that's actually a really good question. So I actually saw Vaccinium mercenites going in a positive trend. It's actually optimum growth is in scrubby flatwoods, and I knew that. But for Vaccinium daroi, I didn't have any predictions for that one. So that was kind of, it is a common scrub species across my plot, but just where my transects were, I didn't get many of that.
Okay, then we have a question about your transects. There's a couple questions. What's the elevation range in your study area? And what interval of sampling points did you use on the transect? And there's a second part, but I'll let you answer that first.
Okay, so can you start with that first one again? Are they asking me like the range?
What's the elevation range in your
from like
study
0
area?
to, yeah, so elevation range is like from 0 to 3 meters from the wetland to the highest rosemary scrub point in my data. That was from 0 to 3 meters. That was my range for that one.
And how far apart were your sample points in the transect? How did you distribute them?
Oh, so they were randomly selected. Like my transects range from like 30 meters to 156, I think was my longest one. So it wasn't like evenly split up. Some of them could be like super close together. Some of them were completely far away from each other. So there was no like standardizing for that one. Those were all randomly selected. Exactly.
Okay, and I have a question for your buddy Haley. Haley wants to know whether you had any thoughts on how below ground biomass might change with relative elevation. Would you expect similar trends or not?
So I would expect similar trends for below ground biomass. However, there's a paper that I didn't include in here, but they tested for that, like to see if like time since fire affected below ground biomass, and it didn't per se, or sorry, it wasn't like exponentially getting bigger. So I'm sorry, biomass did not increase as time since fire increased. Underground biomass did not. So I would expect as relative elevation increased, below ground biomass would increase, just because like of availability of nutrients for the plant species. They will like want a bigger below ground root system. But yeah, that would be actually interesting to look at. I wanted to do that, but below ground biomass experiments are very tedious, and I don't have enough time to do that. I think that would be a PhD, but that's a really good question.
Yeah, I think you're exactly right. You know, our knowledge of what's going on below ground is way behind our knowledge of what's going on above ground.
Yeah, if there was like a regression equation that I could have used to like quantify below ground biomass, I would have definitely did that, but I don't even think that that's the case because it's just so hard to do.
Okay, there's a question from Jim on Facebook, and he's interested in lichens, and in his property, the lichens are only at the higher elevations.
Mhm.
So he's wondering whether you observed any patterns, in lichens with elevation in your study, even though you didn't collect it, whether you saw any patterns.
No, I didn't. That's not something that I looked at, which is actually really interesting. I wonder if that's like related to like soil properties in lichen, because lichen is like, yeah, I did not look at lichen in this project at all. So no, I don't think I would have anything to say about that. I'm actually not, I haven't even really studied lichen often. So I don't actually have an answer for that one. That's a really good question though.
Right. I think that's all the questions, AJ. You did a great job. Thank you very much. And thank you for everybody that attended.
Thank you guys.
Great job, AJ. Thanks again to everyone for attending today. Join us again in 2 weeks for another plant ecology intern seminar. We'll return to our usual Thursday schedule on May 6th with intern Haley Dole. And we'll see you all then.